Welcome to the session of Sexual Reproduction in Flowering Plants Theory Part 2. Disclaimer: the images, text and other material used in our presentations are for educational purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issue, please contact us at sathee@iitk.ac.in and we shall remove the content immediately. And please note that the parts of the content may have been generated using the generative AI, which is experimental. So, sexual reproduction in flowering plants. So, in today's session, our lecture will be based on the pistil, Megasporangium, that is also called as ovule and embryo sac. Okay. The gynoecium represent the female reproductive part of the flower, right? So the gynoecium, in the previous class, we have understood that the gynoecium is present as a female reproductive part of the flower and it is present at the innermost wall, right? The gynoecium may consist of a single pistil. It may have single pistil and that is monocarpellary. Single pistil means monocarpal. One carpel is only there and that is why it is called as monocarpellary. But they may have more than one pistil. But sometimes they can have more than one pistil. So more than one pistil, that is why it is called as multicarpellary. Okay, more carpels are united together and forms the gynoecium part. That is why it is called as multicarpellary. When there are more than one, the pistil may be fused together. Okay, so there are two conditions when there are more pistils, right? When there are more than one pistil, the first condition that all the pistil may be fused together, right? All the pistils may get fused together and that condition is called as syncarpus. That condition is called as syncarpus. But sometime the multiple pistil that is present, they may be free. And that condition is called as apocarpus. So two conditions are there in the case of multicarpellary. Okay, multicarpellary, when all the pistils are fused together, it is called as syncarpus condition. And when all the pistils are free, they are not got fused together, then that condition is called as apocarpus. That condition is called as apocarpus. So here you can see syncarpus pistil. Here you can see this is the syncarpus ovary. All the pistil got fused together. Here you can see that there is one single ovary. It is like one single ovary because all the ovary part got fused together, right? And that is condition. That condition is called as syncarpus pistil. And the example is here, papaver somniferum, right? This is one of the plants. And here we can get alkaloid from this plant that has a medicinal value. So this plant is showing the syncarpus ovary. Another condition you can see, the multicarpellary apocarpus condition. Apocarpus condition, you can see all the pistils are free. They are not. These are the pistils. This is one pistil. This is another. And like this, so many pistils are here. So many pistils are here. It means you can see all the pistils, they are free. They are not united. And that condition is called as apocarpus condition. The examples of the apocarpus condition we see in the case of Michaelia champaka. This is one of the plant, right? Michaelia champaka. So, syncarpus, papaver somniferum, apocarpus, Michaelia champaka. Now, each pistil has three parts. When you see a single pistil, you can see that they are having the three distinct parts. The first part is the stigma. The first part is the stigma, and this is the topmost part, right? The style. From the stigma, there is a stalk-like structure that joined with the lowest part, that is the oval bulbous part, that is called as ovary. So there are three distinct parts in each pistil: stigma, style, and ovary. So here in the figure, you can see this is the stigma part, the stigma that receives the pollen grain. The style part, in which the pollen grain germinate here, transfer its pollen tube through the style, and it reaches to the lowest most part of the pistil, that is called as ovary. Now, ovary is resting upon the thalamus. It is attached to the thalamus. That is the base. Okay. Base at which all the floral parts are attached. All the floral parts are attached. So this is the pistil, and it is having the three parts: stigma, style, and ovary. And this here, this figure is from the hibiscus flower, showing the pistil. Other floral parts have been removed. There are other floral parts also. All the four whorls are there. The sepals are there, the petals are there, and drusiums are there, right? But this is showing the female reproductive part only. Okay, that consists of the three parts: distinct: stigma, style, and ovary. The stigma serves as landing platform of the pollen grain. Okay, the stigma is basically providing a platform on which the pollen grains fall and the pollen grains germinate. The pollen grain germinate on the stigma because it provides some of the chemicals, hormones, okay, that releases the pollen tube out of the pollen grain. The style is the elongated slender part beneath the stigma. Okay, the style, after the stigma, there is a style, and it is the elongated slender part. It is quite a long slender part that comes after the stigma. Okay. And the basal bulged part, is called, of the pistil part, is called as the ovary. It is called as the ovary. And this ovary is very, very important because it consists of, inside there are having ovules in it. Ovules in it, that is the Megasporangium. And that is the main reproductive organ, that consists of the main reproductive organ, female reproductive organ, right? So that is why this ovary is very, very important. Inside the ovary is the ovarian cavity, that is called as locule. Okay. Now, inside the ovary, there is a cavity, there is a locule part. Okay. That locule is called as ovarian cavity, right? Ovarian cavity. And the placenta is located inside the ovarian cavity. There is a placenta. Placenta is a part through which the ovule is attached to the thalamus part. Okay, the ovule is attached to the thalamus part through the placenta, and that is located inside where? It is located inside the ovarian cavity. It is located inside the ovarian cavity. So very, very important point is there that the female reproductive organ, it is having the three different parts: stigma, style, and ovary. Okay, stigma, style, and ovary. The stigma is the providing the basal region at which the pollen grains comes and rest at the suitable temperature. They get the moisture, the hormones, the chemical substances, and that helps to release out the pollen tube from the pollen grain. Now it penetrates the stigma tissue and it goes downward. It goes downward through the style. Okay, style is a slender part. It is an elongated slender part through which the pollen tube goes downward and it reaches to the ovary. And ovary is the lowermost part of the female gametophyte, reproductive organ. Okay, female reproductive organ, and there they are having an ovary. Inside the ovary there is a cavity like a structure that is called as ovarian cavity or locule. Ovarian cavity or locule, we have seen in the case of anther also, they are having the 4 theca. Okay, 4 theca. So in that way also the female reproductive organ in the ovary, they are having an ovarian cavity or the locule. Now, in this cavity only the placenta is there through which the ovule or the megasporangium is attached to the thalamus. Now recall the definition of and type of placentation that you studied in the class 11th. In the class 11th, in the morphology chapter, morphology of flowering plants chapter, you must have studied about the different types of placenta. Basal placenta, parietal placenta, placentation, axial placentation, all the types of placentation you have studied about, right? So, arising from the placenta are the megasporangium, commonly called ovules. Now, this megasporangium, it is commonly called as ovules only, right? This ovule, megasporangia, that is commonly called as ovule. Where these megasporangium are present, they are present inside the ovary only. They are present in the ovarian locule or ovarian cavity. These ovules are present in the ovarian cavity and they are attached. They are attached to the thalamus part through placenta. So that is why this placenta is very, very important. That is the thing through which it is attached to the thalamus, right? So arising from the placenta are the megasporangium, and that megasporangium, also called as ovule, commonly called as ovule, that is present in the ovarian cavity or locule. It is present in the ovarian cavity or locule. Now, the number of ovule in an ovary may be 1. The number of ovules in an ovary may be 1. For example, wheat, paddy and mango. Only 1 ovule in an ovary, right? But to many in the case of papaya, watermelon and orchids. So in these plants, their ovule may contain many, their ovary may contain many ovules. Okay. So this is very, very important point. The examples are very important point. The number of ovules in an ovary may be 1. Only 1 ovule in an ovary. Examples: wheat, paddy and mango. Wheat, paddy and mango. Now there are many ovules in an ovary. There is a condition where in an ovary there can be many ovules present. Example: papaya, watermelon and orchid. So this you must remember. The examples is very, very important. Okay. Now we will see the structure of megasporangium, that is also commonly called as ovule. Megasporangium is commonly called as ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule. Let us study about what is the structure of ovule, how the ovule look like, right? We will study about that today. The structure of ovule. Now here you can see the structure of ovule. This is a diagrammatic view of a typical anatropous ovule. This is the commonest type of ovule in the angiosperm flowering plant. The flowering plant, they are having the different types of ovules, but this anatropous is one of the common type of ovule. So let us familiarize ourselves with the structure of a typical angiosperm ovule. So what you see here, this is a stalk-like structure. This is a stalk-like structure which is called as funicle. Funicle is a stalk-like structure. Now this funicle is attached to the thalamus part through what? Through placenta. They are having the placenta through which this funicle is attached to the thalamus part. This I have already told you. So the placenta is important for the attachment, right? Now this funicle, that is the stalk-like structure of the ovule, is attached to the round bulbous part of the ovule at a point of attachment. That point of attachment is called as hilum. That point of attachment is called as hilum. Now this whole bulbous part here, this is called as ovule. Now inside this ovule you can see there is an embryo sac, and surrounding the embryo sac there is a nucellus. Nucellus is a diploid tissue, right? Now this nucellus is surrounded by a covering like a structure which is called as two covering, inner integuments and outer integuments. Inner integuments and outer integuments. And they are having the two poles. The pole towards this micropyle region. What is micropyle? The integuments here. At this particular point, you can see there is no covering. There is a small pore-like structure when the integuments unite together. So they form a pore-like structure. That pore-like structure is called as micropyle. Now through this micropyle only the pollen tube get enter inside the female gametophyte, right? So that end is called as micropylar pole. That pole or end is called as micropylar pole. Just opposite to this micropylar pole, there is one pole is called as chalazal pole. That pole is called as chalazal pole. So the ovule is a small structure attached to the placenta by means of a stalk that is called as funicle. This I have already told you. The stalk-like structure that is called as funicle. It is attached to the ovule through a placenta. The body of the ovule, the rest of the bulbous part that is called as the body of the ovule, fuses with funicle in the region that is called as hilum. The body of the ovule, it is get attached to the stalk-like structure that is called as funicle. Now there is a point at which this stalk-like structure and the body of the ovule united together. That point of attachment or the union is called as hilum. That point of attachment is called as hilum, right? Thus, the hilum represent the junction between the ovule and funicle. Very, very important. In the examination, it could be asked in a fill up the blanks form of question. The dash represent the junction between ovule and funicle, right? So the hilum represent, the four option could be there and the correct answer will be the hilum. So in this way, you must, when you read, when you study, you must think about from this particular point, what type of question can be asked. It can be also asked, the hilum represent the junction between dash and dash. Dash and dash, integuments and funicle, ovule and nucellus. There could be the four different options, right? But you must remember it is the hilum represent the junction between ovule and funicle, right? Now, each ovule has one or two protective envelopes. Now, we are talking about the body of the ovule. The body of the ovule. The stalk-like structure, that is called as funicle. We have already discussed about. The point of attachment is called as hilum. We have discussed about. Now again, we are now talking about the body of the ovule. This body of the ovule is having the two protective envelopes or covering. It is having the two protective envelopes or covering, that is called as integuments. That is called as integuments. The integuments encircle the nucellus except at the tip where the small opening called the micropyle is organized. The integument is encircling the whole nucellus part. It is covering the whole nucellus part except at the tip. At the tip, there they forms the small opening, and that is called as micropyle. That opening or the pore-like structure, okay, that is called as micropyle. So here you can see, this is the main body of the ovule. You can see the outer integuments and inner integuments, two covering, right? And this covering here, you can see they form a pore-like or the opening-like structure, that is called as micropyle. The pollen tube get enter inside this ovule through this micropyle only, right? Now, opposite to the micropylar end, so where the micropyle is present, that particular end is called as what? Micropylar end. That end is called as micropylar end. And just opposite to this micropylar end, there is a chalazal end. That end is called as chalaza. Okay, that part is called as chalaza, and that pole or end is called as chalazal end. Okay, that represent the basal part of the ovule, and sometime in the examination it could be asked as the basal part of the ovule is called as micropyle, chalaza, funicle, nucellus, right? So the chalaza is the correct answer. That forms the basal part of the ovule. That forms the basal part of the ovule. Enclosed within the integuments is the mass of cells, that is called as nucellus. Okay. Now, the integument, the tissue that is covering the integument, that is called as what? Nucellus. That is called as nucellus, right? Cells of the nucellus have abundant reserve food material. Now, this nucellus cells are diploid in structure and they are having abundant reserve food material. Now, in the case of microsporangia, which layer provide the nutrients? Tapetum layer. Now, here the nucellus is concerned for the storing the abundant reserve food material and providing to the developing female gametophyte. Located in the nucellus is the embryo sac or the female gametophyte. Okay, inside this nucellus, because from the nucellus, one of the nucellus cells get modified and changes into the female gametophyte or the embryo sac, right? It undergoes the meiotic division, the reduction division, and it forms the embryo sac. An ovule generally has a single embryo sac formed from a megaspore. So from where this embryo sac is formed, and it is generally in the single in number, only one, and it is formed from a megaspore. It is formed from a megaspore. Now we will talk about the megasporogenesis, how this megaspore is formed, how this embryo sac is formed. The process of formation of megaspores from the megaspore mother cell, MMC is called as megasporogenesis. So this is again a diploid cell. This is again a diploid cell. From this cell only this megaspore is formed. And that process of formation of megaspores from the megaspore mother cell is called as megasporogenesis. That process is called as megasporogenesis. Ovules generally differentiate a single megaspore mother cell MMC. Okay. The ovule generally differentiate a single megaspore mother cell MMC in the micropilar region of the nucellus. So one of the nucellus cell, it get modified, okay, and it forms the megaspore mother cell. And generally it is formed near the micropilar region of the nucellus. So it is a large cell containing the dense cytoplasm and a prominent nucleus. So one of the cell become distinct from the other cells of the nucellus by accumulating more cytoplasm, and the cytoplasm become very dense, and it is having the prominent nucleus. You can see a very clear prominent nucleus in that particular cell, megaspore mother cell. The other nucellus cell is not having such a prominent nucleus, but this megaspore mother cell become very large in size, accumulate the dense cytoplasm and prominent nucleus. The MMC undergo meiotic division. Now this megaspore mother cell undergo reduction division or the meiotic division. Now there is a question, what is the importance of megaspore mother cell undergoing meiosis? The megaspore mother cell that undergo meiosis, what is its importance? The answer, the importance of megaspore mother cell undergoing meiosis. The first, it produces the haploid cells because it is undergoing which division? Meiosis division, reduction division. So it will later on forms the haploid cells that are called as megaspores. Now that creates a genetic diversity through the crossing over. Okay. Now this, during the formation of this haploid cells, megaspores, they undergo genetic diversity because of the crossing over, because the new recombination is there at the time of crossing over, right? And they create the genetic diversity. They forms the female gametophyte that is called as embryo sac. Okay, they are forming the female gametophyte. Gametophyte means haploid structure. Okay, the embryo sac is formed. Essential for the plant reproduction, and the formation of this embryo sac is very, very important because they are important as they are required for the reproduction process. It enables the seed formation and maintain the correct chromosome number also, because when there is a union of two gametophyte, male gametophyte and female gametophyte, okay, both the haploid unite together and again they form the diploid cells. So they are maintaining the original chromosome number, ensure the species survival, and also facilitate the evolution through the genetic recombination, because in the formation of like crossing over taking place, so there is a mixing of the genetic material, a new recombination is formed, and that helps in the process of evolution. So meiosis results in the production of 4 megaspores, right? There is a meiosis process, and through this process there is a production of 4 megaspore, because one diploid cell when undergo meiosis, it forms 4 haploid cells. So here 4 megaspores are formed. So here you can see this is megaspore mother cell. This is one single megaspore mother cell. The cell, now here these are the nucellus cell. These are the nucellus cell. You can see the surrounding cells are nucellus cell. Now this particular cell becomes very prominent, as you can see the size of the cell. It's quite large. The nucleus size, you can see it's very prominent. The cytoplasm is also very dense. So this megaspore mother cell become very distinct from the other nucellus cell, right? Now it undergo this megaspore cell undergo meiotic division. Here you can see the megaspore dyad is formed, means two cells are formed. Now these two cells again divide and they form the megaspore tetrad. So four megaspore cells are formed. Four megaspore cells are formed, and that is why it is called as tetrads of megaspores. Tetrads of megaspores. Female gametophyte. Now, in a majority of flowering plants, one of the megaspore is functional while the other three degenerate. So we have seen that through the meiosis division or the reduction division, MMC, megaspore mother cell, forms four megaspores, and out of these four, only one is functional. Only one is functional. The other three get degenerated. Right? So only the functional megaspore develops into the female gametophyte, and that is called as embryo sac, right? So only the functional megaspore develops into the female gametophyte, that is called as embryo sac. This method of embryo sac formation from a single megaspore is termed as monosporic development, because only one of the megaspore is involved in the formation of embryo sac. That is why it is called as monosporic development. Okay, such type of embryo sac is called as monosporic development. So here you can see now how the embryo sac is formed. This is very, very important to understand. Now this is one megaspore, functional megaspore, one functional megaspore, and it must be having only one nucleus, right? The one nucleus divide into two, but the division of the cytoplasm is not there, so no cytokinesis is there, only karyokinesis is there. So two nucleus is formed there. Now these two nucleus again divide, and it forms four nucleus, and it forms the four nucleus here. Now these four nucleus again divide and forms total 8 nucleus. So 4 at each pole, you can see they are present. 4 nucleus at each pole is present. Now, what happens? From each pole, one nucleus moves and comes to the center. One nucleus from each pole comes to the center. And in the central cell, you can see the two polar nuclei. These two polar nuclei which are present at the center, you can see they are present as a named two polar nuclei. Now, at the micropylar end, there is one egg cell, and from both the side they are having the synergid cell. Okay. So the two synergid cell and one egg cell unitedly, it is called as egg apparatus. Okay. So only the functional megaspore develops into the female gametophyte, that is called embryo sac, and only one of the megaspore is involved in the process of embryo sac formation, and that is called as monosporic development. And I have here explained how this megaspore, or you can say the embryo sac, is formed. So what will be the ploidy of the cells of the nucellus, microspore mother cell, the functional megaspore, and the female gametophyte? So 4 things are there. They are asking the ploidy. Ploidy, you understand? what is the chromosome number, whether it is a haploid, whether it is a diploid. If it is having the original chromosome number, then it is called as diploid. When it is having the uh, half of the original chromosome number, then it is called as haploid, right? So here you can see they are asking for the nucellus, microspore mother cell, functional megaspore, and female gametophyte. So let us study about the formation of embryo sac in detail. Okay, we will do this, but before that, let us solve this question. So if we talk about nucellus, then nucellus is diploid. 2N, it is diploid. These are the somatic cells of the ovule that give rise to the megaspore mother cell. One of the nucellus cell become very different, become prominent. Okay, they are having the prominent nucleus, accumulate a dense cytoplasm, and it become different from the other nucellus cells, right? So, but basically the nucellus cell is diploid, right? Megaspore mother cell, this is again diploid. Till here, they are become very distinct from the other nucellus cell, but still no reduction division has been started here, right? So also called as megaporocyte undergoes meiosis to form megaspores. Now this megaspore mother cell only undergo the meiosis and forms the megaspores, but still it is a diploid structure. Functional megaspore. The functional megaspore is haploid. Only one of the 4 megaspores survives. The surviving megaspore is called as the functional megaspore because there are the 4 megaspores. Megaspore mother cell undergo meiosis to form 4 megaspores. Out of these 4 megaspores, 3 get degenerated and only 1 survive, and it forms as a functional megaspore, right? Now, the nucellus of the functional megaspore divide mitotically to form the 2 nuclei, which moves to the opposite poles, forming the 2 nucleate embryo sac, right? So earlier the megaspore is having only 1 nucleus. It divide mitotically and the 2 nucleus is formed. Both the nucleus moves to the opposite poles. There is no cytokinesis taking place here. Only karyokinesis, means division of nucleus. Okay. So 2 nucleate embryo sac is formed here. In the figure you can see 2 nucleate embryo sac is formed here. Now these 2 nucleus again divide and forms 4 nucleated states. Now these 4 nucleus again divide mitotically and it forms 8 nucleate stage. 4 nucleus at each pole and it forms the 8 nucleate stage. So 2 more sequential mitotic nuclear division result in the formation of 4 nucleate and later 8 nucleate stage of the embryo sac. We have already discussed about it. And it is of interest to note that these mitotic divisions are strictly free nuclear. It is free nuclear division. Very, very important point. Free nuclear, no cytokinesis division is taking place here, right? So that is the nuclear divisions are not immediately followed by the cell wall formation. No cell wall formation takes place here, right? So it is called as free nuclear division. After the 8 nucleate stage, the cell wall are laid down, leading to the organization of a typical female gametophyte or the embryo sac, right? But once there is a formation of 8 nucleate stage, there will be the formation of cell wall. And here you can see the wall is laid around the egg, the wall is laid around the synergid cell, the wall is laid around the antipodal cells. So here you can see the 3 antipodal cells at the chalazal end. There is a single egg surrounded by the 2 synergid cells. Okay, unitedly this is called as egg apparatus. Egg apparatus. And in the synergid cell, you can see there is a projection like fingers shapes, and that is called as filiform apparatus. In the central cell, now this is the central cell. This is the central cell in which the two polar nuclei are present at the center. So totally it is having how many cells? 7 cell, 3 antipodals, 3 egg apparatus, and 1 central cell. So 7 cell but 8 nucleated structure. The functional megaspore, or you can say the mature embryo sac, is they are having 7 cell 8 nucleate stage. So observe the distribution of the cells inside the embryo sac. What you observe? You can observe the cells, how they are distributed. At the chalazal end, you are having the 3 antipodal cells. At the micropilar end, you can see 1 egg in the center surrounded by the 2 large synergid cells. And these synergid cells are having the filiform apparatus also. 6 of the 8 nuclei are surrounded by the cell wall. Actually, there are 8 nuclei, and out of this, the 6 of the nuclei, they are surrounded by the cell wall and are organized into the cells. The remaining 2 nuclei, called the polar nuclei, are situated below the egg apparatus in the large central cell. In the large central cell, they are having the 2 polar nuclei in the center. There is a characteristic distribution of the cells within the embryo sac. And 3 cells are grouped together at the micropilar end and constitute the egg apparatus. Okay, the egg and the 2 surrounded synergid cells, they are called as egg apparatus. Very, very important point. Sometimes it can be asked, what do you understand by the egg apparatus? So 1 egg and the 2 synergid cells unitedly, they are grouped together and it is called as egg apparatus. The egg apparatus in turn consists of the 2 synergid and 1 egg cell. Now the synergid have a special cellular thickening at the micropilar tip that is called as filiform apparatus. There is a thickening, cellular thickening in the synergid cells, and it is like a projection, fingers like projection, and that is called as filiform apparatus. Though the exact role of this filiform apparatus is still not known, but yes, some of the scientists believe that they play an important role in guiding the pollen tube, because the pollen tube first time get enter in the one of the synergid cell only, and it releases all its discharge in the synergid cells. Okay, the pollen tube into the synergid cells. So 3 cells are at the chalazal end, and those are called as antipodals. Okay, 3 cells are present at the chalazal end, and these are called as antipodals. The large central cell, as mentioned earlier, has the 2 polar nuclei. There is a 1 central cell, okay, and it contains the 2 polar nuclei in the center. So thus typical angiosperm embryo sac at maturity, they are having 8 nucleate and 7 celled stage. They are having 8 nucleate and 7 celled stage. Now we will see the summary. The pistil is the female reproductive structure in the flowering plant consisting of the stigma, style and ovary. Right? So pistil, it is a female reproductive structure in the flowering plants, and it consists of stigma, style and ovary. Within the ovary lies the megasporangium. Okay, within the ovary there is a megasporangium that is also known as ovule. It is also known as ovule, which is the main female reproductive structure that develops into the seed after the process of fertilization. The ovule contain the embryo sac. Okay, each ovule that contain the embryo sac, which develops through the megasporogenesis and megagametogenesis. There is a formation of megaspores through megasporogenesis right? The mature embryo sac typically contain 8 nuclei. If you see the mature embryo sac, it contains the 8 nuclei in the 7 cells, including the egg cell, the 2 synergid cell. Unitedly, it is also called as egg apparatus. It is called as egg apparatus. 3 antipodal cells and 2 polar nuclei in the central cell. During fertilization, one sperm cell fertilizes the egg cell. Okay. During fertilization means in the process of fertilization, one sperm cells fertilize the egg cell to form the zygote, which while the other sperm fused with the 2 polar nuclei to form the endosperm. This complex structure and the process ensures the successful plant reproduction and seed development, which is essential for the continuation of the flowering plant species. So this is all about the today's session, but after that we will discuss about the DPP. So let us solve the today's DPP. So here is the DPP. Let us solve it. The first question is here. Which layer of the microsporangium is responsible for nourishing the developing pollen grains? Okay, we have seen that in the microsporangium, there are certain layer that is responsible for nourishment of the developing pollen grains. So let us see the option for this question. A. Epidermis, B. Endothecium, C. Middle layers, and D. Tapetum. Okay, if we see the microsporangium, the outermost layer is the epidermis layer. Okay, epidermis layer. And below the epidermis, there is a layer that is called as endothecium. There is a layer that is called as endothecium. And below endothecium, there are 2-3 layers of middle layers. Okay, and just below the middle layer, there is a tapetum layer. Now, this tapetum layer, they are having more than 1 nuclei. They may have more than 1 nuclei, right? And they are involved in the process of nourishment, providing nourishment to the developing pollen grain. So, epidermis, endothecium, and middle layer, all these 3 layers of the cells, they are responsible for the protection of the inner developing pollen grain. They are providing protection, right? So that could not be the responsible for providing nourishment. Yes, but the tapetum layer is there for providing the nourishment. So we will opt this, save, and move to the next question. The next question is here. In angiosperms, a mature male gametophyte derived from a pollen mother cell has how many cells they are having? They are having 2 cells, A 2 cells, B 3 cells, C 4 cells, and D 5 cells. How many cells are there? So there is a first meiotic division of the cell and it forms the 2 cells. And later there is again second meiotic cells and they form the 3 cell stage, right? So I'll opt for this 3 cell stage because it is not 2 cells, 4 cells or 5 cell stage. It is 3 cell stage. So we'll save and move to the next question. What is the ploidy of the functional megaspore in a typical angiosperm ovule? Ploidy of the functional megaspore. Now the term is there. If it is all megaspore or functional megaspore, right? So let us see the options here. Haploid N, diploid 2N, triploid 3N, and tetraploid 4N. So this is the ploidy in the option. Now this functional megaspore is formed by the division that is called as reduction division or the meiosis division. Reduction division or the meiosis division, right? So what is formed after? What is the ploidy of the cells that undergoes the meiosis? Haploid cells are formed. Okay. So here also the functional megaspore is formed through the meiosis division, reduction division. So it will form the haploid cells. So we'll save and move to the next question. Which structure in the embryo sac guides the pollen tube to enter the synergid? There must be some structure in the embryo sac that guides the pollen tube to enter to the synergid, right? So what is that structure? You have to find from the given options. So let us see the option here. A, antipodals. B, central cell. C, filiform apparatus. And option D, egg cell, right? So in the synergid cells, there are having some outgrowth, finger-like projections are there. That is the cell wall growth, and that is responsible for guiding the pollen tube to get enter inside the synergid, and it also receive all the content from the pollen tube. Okay. So that particular structure is called as filiform apparatus. Filiform apparatus. It is not antipodal cell. It is not central cell. It is not egg cell. It is filiform apparatus. So we will opt this filiform apparatus, save and move to the next question. So the next question is here. In artificial hybridization, emasculation is done too. Why the emasculation is done? A, protect the stigma from contamination. B, remove the anthers to prevent self-pollination. C, collect pollen grain from the male parents. And D, rebag the flower after pollination. Right? So in artificial hybridization, why the emasculation is done? So that the anther, remove the anther to prevent the self-pollination. There should not be the self-pollination. To avoid self-pollination, the emasculation process is done. So we will opt to the option B, right? And move to save it and submit it. Then we'll see the result. Okay. Yes, all the answers are correct. Right? So this is all about today's session. Thank you very much.